It's a fascinating subject. I'm delighted to be welcoming to the virtual stage Miguel Toribio-Mateas. He's going to be looking at the impact of plant-based meat alternatives on the gut microbiota of consumers. Hi, Miguel. How are you?
I'm very well, thank you. Yes, and yourself?
Very, very well. Delighted to have you with us. It's been a while since we spoke, but I know you're an absolute expert in these areas, and your presentations are always absolutely fascinating. I love watching you present. So delighted you're able to join us.
So no pressure at all then?
None at all, no. And you've changed location to where you usually present, actually, but
yeah, I have changed just because my dogs are quite noisy, so I've been hiding from them slightly. Yeah.
Thank you so much, Miguel. I'm gonna pass over to you. I think you should be able to share your screen now.
Yeah, I'll do that. I'm going to do that right now and actually go and do. So you should be able to see my slides now.
Yes, we can. There we go. Okay,
yeah,
I am going to hand over to you then. Over to you. Thank you so much, Miguel. I'm absolutely excited for this.
Thank you. Just to make sure that you see them moving, can you just give me a nod that you see them moving?
Yes, we see them moving.
Yeah, okay, brilliant. Okay, so I'll go back to the beginning. Excellent. Okay, well, thank you for having me. I'm Miguel Toribio-Mateas. I'm a researcher at the School of Applied Sciences at London South Bank University. And today we're talking about the impact of plant-based meat alternatives on the gut microbiome of consumers. And the study is quirky. I'm going to introduce the rationale first. And because through COVID, we have realized of many things, that we need to move more, that we need to eat more healthily, that diet and food choices are actually not isolated, that they have an impact, not just on ourselves, but also on the well-being of others and the well-being of the planet. So there's an interconnected kind of like nature that we have identified through the pandemic that has been quite interesting to observe, actually. And public health messages of this nature that goes beyond our own personal health have actually made us choose foods that are the kind of foods that we're talking about in the study. So there's been a huge increase in flexitarianism, for example, which is this kind of a lifestyle behavior where people are not full vegetarian or vegans, but they are more attracted to replacing some of the animal product with plant-based products on the basis that they understand that they have an impact, again, not just on themselves, but also on the planet. Ultimately, there's a lot of talk about planetary health and all the spectrum of health that is in between us as individuals and the planet. So from public health, which is obviously a very well-known kind of like construct, to global health, to One Health, which was popularized by a few papers published probably 2016-17, and we've moved on to the Lancet planetary health kind of initiative, which everybody seems to be talking about all the time. So that actually gives us a lot of good reasons why people should understand that a plant-based diet or a plant-based lifestyle is a good thing, whether it's fully plant-based as in vegan or plant-based as in substantially based on plants when we make food choices, but then some smaller amounts of animal products as well added in there. But that should give us the rationale really to understand that the plant-based market makes a lot of sense in 2021, 2022, not least because Bloomberg actually described the market as explosive in growth only in August this year. The features that are published every day in websites like Food Navigator, Nutrient Ingredients, is really big websites that everybody reads, and blogs, massive big blogs like Green Queen, Plant-Based News, all of these are basically making us understand that this is a really big trend and that flexitarians are actually driving the trend to a great extent. So it should actually be quite easy to understand that the reasons why people are choosing these foods. Yet we look at the foods in the supermarket and we see fake meat. I actually don't really like the name fake meat. A lot of people use it. It was used a lot to refer to the study as well. So we see fake meat, and it makes me think of two things. Well, it makes me think of the evidence. So the evidence that is available in literature tells us that when people who are confronted with the choice of a substitute for real meat, animal meat, are given a plant-based meat, they don't like it to look plant-based particularly. They don't like it to look like it's made of lentils. They like it to look and feel and taste like a real, like the original product. So if it's a burger, they like it to look, taste, smell a bit like the original burger. And there's also some evidence suggesting that people who are either flexitarians, vegetarians, or vegans have more of a tendency to consume foods that are classically thought of as being ultra processed, as in the NOVA classification. So really kind of like long shelf life, additives, ingredients that you may not have thought about adding to a burger, maybe in a plant-based burger. So this gives us really an interesting paradox in terms of the study. So we have all of this evidence actually suggesting that plant-based foods are good for us, and the consumers driving that growth in that market are actually choosing foods that might be not so beneficial for us because they're ultra processed. What is this about? So what is the nature of ultra processed foods for the microbiome particularly? So bringing it to the core of the subject today, it means that there are foods that, because they have been processed more than they might need to be processed, they are going to be digested in the upper part of the digestive system. So when it comes to colonic bacteria getting a substrate to feed themselves on, then there's very little there for them to enjoy. So that is a problem. So if you have refined carbohydrates, for example, or excess sugars, they're going to be mostly metabolized, absorbed in the small intestine. There's going to be very little getting to the colon for them to, for Faecalibacterium or Rosaburia, for example, to feed on. So that is a bit of a problem. And we were really curious as well as scientists to find out what happened when people actually ate these plant-based meat alternatives, because we came to the study completely, big disclaimer, we were not fervent vegans trying to prove that vegan meat is better than animal meat. We came to the study completely with a neutral scientific hat on. We were not paid by the company that I can name, is Meatless Farm, to conduct the study. They donated the product for us to use, and we came to the study with no ideology. So we didn't want to prove that animal meat is better, or that vegan or fake meat or plant-based meat is better at all. So we put together the study that is explained in the graphical abstract. So we basically split 40 participants into two groups. Gender split was pretty 50-50. And the ages were ranging from, I believe, 21 to around 55. So that was kind of the age range. And one of the groups was actually asked to replace around 5 meals a week. So it wasn't really prescriptive. And this is one of the things that some people believe is a bit tricky in terms of the limitations of the study. And I can understand it is a limitation, but we were trying to emulate the behavior of a consumer who actually goes to the supermarket and decides, okay, well, today I'm a flexitarian, I fancy replacing my regular burger with a plant-based burger, as opposed to prescribe it and control it in such a prescriptive way, for lack of a better word. So it was basically allowing them to replace as many or as little meals as they wanted. And the average, some people replaced lots of meals. So there were a couple of people who replaced like 14 meals a week. Some people replaced only 3. The average was around 5, which is over 5 meals in a week. The other group were just asked to continue with what we call a typical British diet, which is basically, they were selected because in the inclusion protocol, they told us that they ate animal produce every day. So whether it was meat, fish, sausages, eggs, you know, every day they were consuming these products that came from animals. So that was what they were doing, and ethically there was no problem with them continuing doing what they were doing. So that was the rationale. And basically we went through the screening, we selected the participants, we split them. That's the kind of way that we did it. And then we also selected a method. So we needed to put, you know, I showed you before, we did before and after stool test. That was the main kind of like chunk of the method that we based our understanding of what happened on. And we use 16S sequencing. So there's advantages and disadvantages in 16S. Is widely available. It's gives us loads of insight at the genus level. It's not as precise as shotgun sequencing in terms of identifying with precision the species or even, you know, strains. Forget about it. But you have a really good understanding at a pretty good level when you have good enough reads what is going on. We want to see, you know, is there any shift, and without spending, you know, hundreds of thousands of dollars that we didn't have, because I'm working in a unit that is it runs with a grant. We haven't got tens of thousands of dollars or pounds to do these studies. We run on a shoestring. I wasn't paid for the hundreds of hours that I was spending doing the study, and so it's we were doing it out of the kindness of our hearts, just to understand what goes on when somebody eats this kind of products. So we use biostatistical method that is called is described as compositionally aware, which basically means you every time that a taxon actually changes, whether up or down, we analyze the level of change in relationship with every other taxa that has actually changed as well. And we make sure that the relationship is relevant using a statistical method that is an adjustment post hoc called FDR, which basically tells us whether we need to give it a lot of attention or whether it's actually not non-significant. So, and we did that within groups and also between groups. So I also need to show you what the characteristics are of the products that we were giving these participants. So we have basically had a product that, when you look at the breakdown in terms of the macros, is high in protein, vegetable protein, is high in fiber. So those are the kind of like main highlights. And then if you look at the micros, if you look at the micronutrients, you have an interesting profile of polyphenols that might also be behind the changes that we observed. So what did we find? And I'll just have some tea because I've got really dry mouth. Okay, so we have some nice changes that you can observe here between the intervention and control. You start seeing some changes in butyrate producers, so Ruminococci, Lacnospiraceae, Oscillospira, Rosaburia. So those are at the kind of like either family or genera that have actually changed. So major taxa changes, and you can see the zero there and the rate of change at the bottom. So nice changes, as well as some parallels between the taxa that had actually experienced change. So we found relationships that were actually significant between some the changes in some taxa. So within group differential abundance analysis between Lacnospira and Faecalibacterium, for example, found an interesting relationship in there. So they seem to move at the same time. They were almost like holding hands. So when the Lacnospiraceae, or the Lacnospira in this case, so a little bit lower down, the genus of the Lacnospiraceae family was going up, the Faecalibacterium was also going up. So interesting insight in there. Both are responsible for the production of butyrate, which is one of the main insights. So very interesting insight in there. And very same thing, of very similar thing with the ruminococci and oscillospira as well. So a relationship in there again between a family and a genus that again is involved in butyrate production, which is quite an interesting insight. We also found movement in the butyrate synthesis pathways. So again, because we're dealing with 16S data, we're not looking at metagenomics or metatranscriptomics to actually understand what precisely the amount of butyrate that was produced. So this was an approximation based on advanced bioinformatics that our colleague Natalia Klimenko was able to figure out. I have to admit, a lot of them go completely over my head because these bioinformaticians that can actually do everything from scratch in Python, I'm in awe. So I can just about manage to explain what the method was about. But it's an approximation based on the data that we get from the sequencing, from the 16S sequencing. But based on that, we see that there are changes that are just about significant in the 4 butyrate synthesis pathways, but particularly on the glutarate and the aminobutyrate succinate pathways. So those are the key ones that were so close to significance that it was, very, splitting hairs whether it was a p-value that was significant or not. Very, very close to that. So that was very interesting. And then we also had some changes interesting changes in alpha and beta diversity. So the alpha diversity didn't change to an extent that was significant either in the intervention or control. And I was a bit surprised actually, because this is the kind of thing, because diversity is something that's very close to my interest as a researcher in the microbiome field. I was expecting that those people who were giving up some animal protein to replace it with vegetable protein and fiber and augmenting the level of fiber by about just under 20 grams in a week, which isn't a huge amount of fiber, but if somebody's fiber depleted, it's better than nothing. I was expecting the diversity to go, the alpha diversity to raise in the intervention versus the control, but that didn't quite happen. It was very, it wasn't quite significant, but we did observe some better movement in terms of the spread of the diversity in the beta diversity score. So that was, in terms of Hutchinson distance in the diversity score. And then we have an interesting move as well in phylum, which is the Tenericutes, which is relatively unknown. It's not something that we have a huge amount of literature on, particularly in the realm of dietary approaches to modulation of the microbiome. So of course, there's plenty of literature about Tenericutes, but not so much as about how we modulate them up or down with diet, and particularly in humans. There's a handful of studies. We have a handful of studies in animals with walnuts, for example, being given to rabbits and mice, but there's very, very little evidence that Tenericutes actually can be modulated at all with diet. And we found that they tend to be higher in meat eaters anyway, and they tend to contain, again, because we're talking about unspecified Tenericutes, we didn't get to the bottom of which ones were high or low. We're talking about bugs like Mycoplasma or Uroplasma that in general, they don't serve a positive purpose to health. So we conclude that if the Tenericutes actually went down in the intervention group, whereas they actually went up in the control who were eating meat, continuing to eat meat, we tend to see that as a positive observation as well. So in general, what we saw is a small but significant change in butyrate production in terms of the relative abundance of butyrate producing taxa. So the Faecalibacterium, the Rosaburia, Eubacterium, Ruminococcus, Coprococcus, Eubacterium halley, and so on. And then a reduction of tenericutes in the intervention group, a small improvement in beta diversity in the intervention group versus the control, whereas we actually didn't see any of these changes at all in the control. We only saw an increase in tenericutes. So that was the only thing that we saw that was significant to highlight. And obviously, diet is not the only factor that affects the microbiome. We know that, geography, time of the year, it was veganuary, so people may have had a bit of a health kick in the control group and decided, okay, well, I wasn't supposed to be changing anything, but it's January, so I want to change the way that I eat a little bit because, my husband or my partner or my flatmate is doing it as well, and I feel a bit embarrassed not to join. There's obviously age and, all of these other factors that we cannot control, how we were best breastfed and birthed and so on. But diet is definitely a really massive factor affecting the microbiome. We let the consumers actually manage how many of these meals they wanted to replace, because we wanted to make it as real world as possible without prescribing the exact amount of meals that they had to replace to abide by a protocol. And basically, we were able to confirm that plant-based meat alternatives, when replacing animal protein with these products like the burgers and meatballs and so on that we replaced in the study, they tend to they contribute to an environment that is more akin with eubiosis as opposed to dysbiosis. And it was interesting to observe this. Obviously, it's a small study, it's a short duration, it's a parallel control trial, so no crossover, because we don't want to lose loads of participants with, with 20 in each arm. It's difficult to do a crossover and actually end up losing another 10. So the conclusions really for us were that plant-based meat alternatives might be ultra-processed in some cases. People might see them as that. They go to the supermarket, they look flashy, they might look a little bit, they are plant-based, but they might look a little bit artificial, and they may have a judgment about them. But in general, used as part of a balanced, healthy diet, they actually can play a role in somebody's health. And they're obviously, good for other causes as well, depending on the ingredients, obviously. But if they are sustainably sourced, they can actually play a role in planetary health and so on. So it was an interesting study. Hopefully, we'll be able to take it forward with doing, shotgun and long gun crossover and so on. So I wanted to thank Nathan and Zoe, as well as my colleagues involved in the study, and all of you for listening to me today. And I'll be glad to answer any questions that you have for me. Thank you.
Thank you so much, Miguel, and thank you for the thank you. We're delighted to have you here, and we've got so many questions coming in. It's going to be interesting to try and order them. Let me have a look. I think a good place to start would be to ask you about some of the study design. We've had several questions focused on that. The first one I'm going to kick off with is a question asking about what was part of your inclusion criteria when selecting participants. With the low budget that you had that you mentioned, was the inclusion criteria quite limited, or is there anything you can talk to us about that?
Well, I think rather than actually talk about the inclusion criteria, I think the exclusion criteria were quite strict.
Yeah.
So we basically had, I'm just reading, anybody with an immediate DNA family with a diagnosis of any IBD, IBS, or bowel cancer, they were completely excluded. Anybody allergic to any of the ingredients, anybody with a history of mental health or brain cancer, anybody diagnosed with any condition for which they receive NHS support. So that basically screened out anybody, who is taking a tablet for anything with a prescription from their doctor, whatever that anything might be. and anybody who had a positive COVID-19 diagnosis or suspected COVID-19 symptoms in the previous 6 months, just because we know that COVID can actually alter the characteristics of the microbiota composition in people who have had the condition. So yeah, so I think, you know, if I were to just go quickly through the inclusion criteria, so no underlying health conditions, age between 18 and 55, just because before 18 you get into, you know, puberty, and, like, you know, it's a mess in the microbiome with like hormones going up and down. Over 55, then you start getting into comorbidities that make things difficult with this kind of like small sample. A BMI that was restrictive as well, so somebody who was not too slim or too overweight. No antibiotics in the past 6 months, no probiotic supplementation in the last month, and eats meat, poultry, fish, eggs, or cheese daily, and does not eat plant-based meat or substitutes. So basically, somebody who was eating these kind of products on a regular basis was excluded straight away. So we had a huge interest. I think it was the study that was the most popular in terms of screening. I think we had something like 300 plus people contact us. A lot of work to screen out. So it was my colleague Adri who was in charge of that. So she did a huge amount of work in screening.
Excellent. We've also had a question on how you measured things like the short chain fatty acids, and whether the people in the study had to keep things like food diaries, whether you provided recipes that people had to follow, or whether you just sort of left them to it and that sort of thing. Again, all part of the study design, I guess, and the methodologies.
Yeah, so the study design did offer some support with recipes. So Meatless Farm has a nutrition consultant who worked with us and created a booklet that had loads of different ideas to incorporate different products. So it wasn't just left to them to decide, you know, I'm going to eat meatballs today or just tomorrow. There was kind of a, you know, a structure for people to follow if they wanted. In terms of the analysis of the short chain fatty acids, you can actually measure the short chain fatty acids with a specific method for that kind of a measurement, but it's costly. You need a different bottle to send, you know, as an additional sample. It ideally needs to be refrigerated, and it, you know, if you send it in the post, it kind of like perishes really quickly. So we basically had an estimation. So the estimation of the potential for synthesis of butyrate as well as acetate and propionate, which were not anything to write home about, is based on the taxa and on the relative abundance of each of the butyrate producers in the control group and the intervention group, as opposed to the actual measurements of short chain fatty acids. This is one of the limitations that we could go about by doing, you know, an extra, taking a sample and sending it off and doing it through another method. But yeah, in this case, we wanted to make it as easy as possible for people to comply, because microbiome studies in the real world, people are all up for them. And then when it comes to actually complying, you lose a lot of people because they don't know how to do the sample, or they didn't expect that the sample was going to be so messy to take, or, you know, so if you ask them to do 2 or 3 bottles as opposed to 1, you end up losing a few participants that could be quite valuable.
Sure. And can you remind me as well, how regularly the samples were taken? What was the duration?
So we just did 2 samples. It was really straightforward design, you know, split them into 2 split them in terms of age and gender into 2 groups that were 20 and 20. We only lost the 1 participant who basically had to take antibiotics through, you know, some dental infection or something. So, and they just did that before and after. So basically everybody was asked to do a sample within a window of a couple of days, so within 24 hours of each other. So everybody complied with that. And then everybody was asked to do the second sample again within the same 24 hours, just in case there was a bug going around or anything. So it was a way to control that kind of environment.
Okay, great. Thank you so much. We've got a couple of minutes left before we have to move on to our next presenter. One of the other questions that we've been asked was whether you did any analysis for different strain types, or whether it was based purely on sort of general taxa and families. So whether you looked at specific bifidobacteria, lactobacilli, and that sort of thing, whether you saw any changes in those.
So we did look at lacto, bifido, and opportunistic bacteria as well. Your typical suspects, Citrobacter, Klebsiella, Pseudomonas, Campylobacter, whatever. There was no changes in those, so there was no antimicrobial effect, so to speak, of either the control or the intervention, and there was very little movement in terms of the lactobacilli or bifidobacteria as well. We didn't, so we did look at those aspects of the microbiome, but with 16S, even with a huge amount of reads, even if you go over, completely over the top and you have 150,000 reads or 170,000 reads, if you have the machinery, it's very difficult to identify with precision the strain is impossible with 16S. You need a shotgun to do that, and so yeah, it's quite tricky to do that with 16S.
Sure. Okay. Just in 30 seconds or less, if you have one thing that you want people to remember, just to round us off, what would that be, Miguel?
Yeah, so the study had loads of interest from the press, which is fantastic. I would just say that because we based the study on products from one particular company, the assumptions that the products can be good for the microbiome, they cannot be completely generalized to all potential brands. So I don't know whether another brand is going to do the same. So ideally, we would need to look at a mix and match of different brands and actually compare them. And I don't know how much appetite there would be from brands to do that, but that would be the ideal scenario. Otherwise, we're assuming, we're generalizing that all brands of plant-based meats are possibly good for the microbiome, but some of them may not be.
I'd love to see an arm with an unprocessed sort of.
That would be good. Yeah, if people were up for it, it would be ideal.
That sort of maybe three-way split between meat, sort of the processed meat replacer, and actually a sort of a whole food plant-based diet. Anyway, I digress. Thank you so much, Miguel, for your presentation. Really interesting. We've had so many great questions, so much great feedback. Lots of love for you in the room. Unfortunately, that is all we've got time for, though, because we do have to move on